Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about prokaryotic DNA replication. So mainly, I'll focus termination process. So before going to have the main discussion, we should know that what are the required protein and enzymes for termination process. So DNA polymerase 1, 2 and 3, these three proteins are required for elongation as well as the termination steps. And DNA polymerase 3, it only can add nucleotide from 5' to 3' direction, and other two enzymes, they have the exonuclease and proofreading activity and DNA repairing activity. And along with that, terminator site or in short TAR site and TAS proteins are also required to arrest replication fork. So let's have a look at what are the terminator site or TAR site. So basically the TAR site has been found in the terminus site of the circular DNA, and it has been found that in E. coli it has the 10 TAR site, and each site having the 23 base pair long DNA bases, and those are denoting TAR A to TAR J. And also TAS protein is very essential to form a complex with the TAR site in order to arrest the replication fork, and each ter-tus complex having the one permissive phase and one non-permissive phase. So as you see here, this is one permissive phase and another non-permissive phase. So replisome or DnaB helicase can pass through from this permissive phase but cannot pass through the non-permissive phase. So as you see here in the E. coli, they have the 10 TAR site: TAR J, TAR G, TAR F, TAR B, TAR C, TAR A, TAR D, TAR E, TAR I and TAR H. So the replisome, it can approaches from ODC to the terminus site through the permissive phase, but here this both of the fork fuse together and trap here, because here is the non-permissive phase and it cannot go further. So here, when it comes here to forks, then it means that new DNA synthesis is complete. So next, have a look at how this ODC it propagates, and this how this DNA replication fork it propagate from the ODC. So as you see, this replication fork, it's approaching towards the terminus site, and there are two replication forks. So always remember that in one circular DNA contain only one bubbles, not more than that, and one bubbles means two replication fork. So two replication fork, it approaches in two different directions, so it is the bidirectional. And then so it comes across through this permissive side and also this permissive side. So when the new DNA synthesizes completely, then it trap here by the non-permissive phase. So let's have a look at, and also there is the different TAR site sequence. So this is the 23 base pair long, and this shaded region are indicating the TAS protein bind here. So TAS protein contain on one DNA binding domain. So TAS protein can bind in these DNA bases. So let's have a look also that another the sequence and secondary structure of the TAS protein. So this is the amino acid residue of the TAS protein. So this blue color, it's indicating the non-specific contact with the TAS DNA, and this red color amino acid residue, it's indicating the direct contact with the TAR DNA. So let's have a look at the termination process. So here, as you see in this figure, that there are in this bubble like replication fork 1 and replication fork 2. These are approaching towards the terminus site, and in this terminus site these two forks fuse together. So here this is the replisome or DnaB helicase, as you see here, and this green color, it's showing the DNA polymerase 3. So the replisome or DnaB helicase approaches towards the terminus site through the permissive phase of the ter-tus complex, and upon passing through the permissive phase of the terminal ter-tus complex, DnaB helicase cannot go further, so its action blocked by the non-permissive phase of the terminal ter-tus complex. So then the helicase, it breaks the in-between hydrogen bond and it unwind the DNA. So then the DNA polymerase 3, it polymerize the remaining unwind DNA strand, and in the leading and lagging strand as well. So next step, so when the replisome or the DnaB helicase, it approaches near the terminal ter-tus complex, and its action is blocked by the non-permissive phase, and then the in between the remaining hydrogen bond is breakage of this hydrogen in between hydrogen bond is occurred by this helicase, and then in the terminus site the two replication fork fuse together. And next process is the DnaB helicase assist primase to lay down the last lagging strand primer, and then this the lagging strand synthesize. So then the DnaB helicase action is blocked by the ter-tus complex and then dissociate from the template, and then the last unwind DNA strand, it's polymerized by the DNA polymerase 3. So DNA polymerase 3, it complete the leading strand synthesis, as well as also the last lagging strand synthesis, and then the RNase H and DNA polymerase 1 remove the primer and cleave the RNA primers, and then DNA polymerase 3 fill the remaining gap that occurred by the removal of the RNA primer, and then the DNA polymerase 3 dissociate from the template, and then the ligase, DNA ligase, it ligate the nick in between the new strand by forming the hydrogen bond, and the final step is the DNA polymerase 2 and 1. It can recognize the DNA misbase pairing and repair those mispairing. So this is all about the termination process. I hope this video will be helpful, and if you like this video, kindly hit the like button, share it, and subscribe my channel. Thanks for watching.